Bonjour à toutes et à tous. Tout d'abord, oui, je dois dire que je vais faire ma présentation en anglais parce que je ne suis pas au niveau déjà en français. So good morning all. Maybe I first introduction to myself. So my name is Ross Jeggo, Englishman, been in France now 4 years working at Servier. I am the head of the neuroscience and immuno-inflammation therapeutic area at Servier for the last year. I'll talk a little bit more about that. I'd just also like to say thank you to Philippe, Jan, Fabrice and the Oncodesign team for the introduction to come and talk to you today about our exciting partnership. So just a brief background to Servier. I'm sure you're all very well aware of Servier, but I thought it was an important opportunity to introduce them, of course, to those that don't know so much. It is a unique pharmaceutical company in that we are governed by a non-profit foundation, which gives us certain advantages in terms of our mid and long-term perspective versus shareholder ownership. We're guided by a long-term vision, and we invest a lot in R&D, which is good news for me, of course, in R&D, and allows us also to take different, I think, choices along the way to invest and grow our portfolio of promising opportunities for patients. One of the facts we're most proud of as a company is that we treat over 100 million patients worldwide across 150 countries, so we have a big geographical breadth and presence. We have an important strategic ambition to become a recognised and innovative player in oncology, clearly not in my direct domain, but something very important to Servier, and perhaps some of you have seen the acquisition of Shire a couple of years ago and Agios in the last year. Strong R&D capabilities across a number of different research institutes, Orléans, Paris, the US as well, now with Agios. More to come on that at the very end of my talk, but we also have a very broad clinical development centre which allows us to access obviously a diversity of patients that's super important for the ambition we have in clinical development to bring new medicines forward. 22,000 employees across the world, so it's a mid-sized pharma, but again, a broad geographical presence. Just some financial numbers to give a background, but our total revenue 1920 was 4.7 billion. With a decent EBITDA, again, we've put that back into the business. 70% of that coming from Servier brand name medicines and 30% from our generic business. So moving more into my domain, just to give you a flavour of where this project and partnership sits. As mentioned, neuroscience and immuno-inflammation. A strong background in neurology at Servier. We have an antidepressant on the market, Valdoxan, and a number of other projects, Trivastal, Stablon products, Stablon and Arcalion. Immuno-inflammation is an up-and-coming area, I would say. We have some products in clinical development. We have a very highly focused disease biology strategy. It has to be said, we are a mid-sized pharma. We invest heavily in oncology across neuroscience and immuno-inflammation. The investment is very, very precisely centred on specific indications. Across the neuroscience domain, we have a phase 3 program, bumetanide, for autism spectrum disorder. We get the results this summer. We're all very excited about that. Promises to be a game changer to target the core symptoms of autism. We also have I would say a growing discovery portfolio across movement disorders, rare neurodegenerative diseases, up to Parkinson's disease, which I'll talk to you about today, and actually a particular population of Parkinson's disease. We have a biological focus on protein clearance mechanisms and particularly RNA targeting, which I'm sure we're all familiar with of course the COVID vaccines that have come through that technology. Across the immuno-inflammation, not so relevant today, but just to say a few things, we have a couple of clinical stage assets, a growing discovery portfolio, an interest in Sjogren's disease, but also some more targeted autoimmune indications. Again, thinking about precision medicine approaches and how we can bring, with a higher feasibility, a higher probability of success, products through to clinical success. A particular biological focus across autoantibodies and adaptive immunological pathways. So, it's maybe overselling it, but it's important to state it. I managed to find the new logo as well, so that's a good start. But it's a true partnership, and this was first introduced to me in 2018, shortly after I joined Servier. I was aware of Oncodesign before that, and of course Servier and Oncodesign have worked for much longer than that. But that's where I came to know the Oncodesign team, And in particular, we had an interest in LRRK2, a target that I've been interested in for a long time. In my former life at another pharmaceutical company in Denmark, and before that as well. A very, very, very challenging target. It's important to say that, because I think actually what we've announced in the last week shows the huge progress we've made through this partnership. So we launched the research collaboration in March, just a couple of years ago. And again, it's I think, achieved a lot in a short period of time. Both bringing together rich and diverse experience between the two teams, the chemistry experience around the nanocyclics and some of the biological experience that we have at Servier, to really bring forward a product or a drug into hopefully clinical development next year that promises a lot, again at a very, very challenging domain. I've put agility at all stages. I think that's really important actually, and particularly with COVID upon us. The man walking the tightrope is supposed to represent the VUCA world that we all know: volatile, uncertain, changeable and ambiguous. But it's meant, that we've been able to balance resource commitments. There's been obviously differences in some of the lockdowns across the geographies of France and the world, as we know, and that's it's been difficult, I will say, to maintain drug discovery and development across that changing and difficult environment. And I recognise the agility that has been shown by the Oncodesign team in answering the needs of the project as we've continued on our ambitious trajectory to get this product into humans. A little bit about the disease we're targeting. It is, of course, Parkinson's disease. More than 10 million people globally living with Parkinson's disease, so a huge unmet medical need. There are no disease-modifying or disease progression-altering treatments available today. It's a progressive neurodegenerative condition, 1% of the population over 60 years old, more men than women, interestingly. The symptoms, of course, I think those of you who know Parkinson's disease is dominated by motor function impairment, but actually there are many other very important quality of life challenges. One of the first signs of Parkinson's disease is anosmia. You lose your sense of smell, and postural changes as well, and many others. As mentioned, there's nothing that addresses the disease progression, but there are drugs out there that treat the motor symptoms in particular, levodopa being the most famous, but others as well. They have a limitation both in the effect duration that they have and the side effects that you get from them. So again, the important message here is it's a very, very, very large unmet medical need. There are no treatments available to alter the disease progression, and that is what we are bringing forward this partnership to try and address. That's an interesting change in the slide, but hopefully it'll work. Maybe there's an animation. Okay, so getting onto the target, leucine-rich repeat kinase, a kinase enzyme, a big one. So it's 288 kilodaltons for those interested. That's a large molecule. The function of it, the precise function of it, still debated in the field. Many publications. It's clearly not a single function. It has a diversity of activities. Many of them centered around autophagy, protein clearance. So you can remember from my earlier statement of our interest biologically, that is at the center of it. Macroautophagy, chaperone-mediated autophagy, and lysosomal biology. These are some of the functions known that LRRK2 plays a really important role in. Why have we been interested in this target? For me, I think the field, particularly in neuroscience, is littered with failures for a number of reasons. It's a complicated disease, it's the brain, it's difficult to get drugs in there. But also because we've actually not been able to adequately answer the questions that we've tried to ask in the clinic. So measures of target engagement and understanding the validation of targets is somewhat lacking in this field. And that's where LRRK2 brings forward a strong advantage. There is GWAS, genetic-wide association study, evidence heavily implicating this target in the disease biology. So the graph in the middle is intended to illustrate that. There are a number of mutations in this kinase that confer an increased likelihood or risk of developing the disease. It is thought to be a toxic gain of function of the kinase that is associated to the disease. G2019S, a glycine 2019S serine. So this is the location on the kinase, is the most common and well known, and there are specific populations around the world that have a high proportion of this mutation in their populations, Ashkenazi Jews, Berbers in North Africa. There are a number of others as well across Asia, and the contribution to sporadic idiopathic, i.e. without a cause, Parkinson's disease is not known but is expected to be significant. Interestingly, there is also a mutation that we think drives a loss of function that is actually protective of Parkinson's disease. Again, all that gives us is the confidence that this has a contribution to the progression of the disease, so validating the target. Lastly, on the right-hand side, translation. So this is important because, again, in terms of accurately asking the question in the clinic, we can use good markers of kinase activity, either phosphorylation directly or surrogate measures. RAB10 is one that's been identified relatively recently, to give us an idea of the impact of inhibition of our drug on the function of that kinase. So far today, these are easily measured in the CSF and in plasma. Easily is a relative word, but the measure in the brain is more challenging. I'll perhaps come on to that as well as time allows. As mentioned, they are genetically identified subpopulations of patients, so it allows us to take that precision medicine approach to be able to target the patients that we think will benefit most from this treatment. It's important to say that LRRK2 is not just in the brain, it's throughout the body, so there are some effects of inhibiting this kinase with a drug that are useful in some ways. They are a marker of activity. They cause pronounced phenotypes, but there is no known toxic result of this activity outside of the brain today. So where are we today? I think it's been said a few times already, but we have selected our first preclinical candidate, which is honestly a fantastic achievement. We had a pretty high bar in terms of the quality of that candidate, based on the field, based on the experience of what we need to achieve to make a good drug. And it looks like we have a very, very promising candidate that we're taking forward to human studies. Very potent, importantly, which again has been a challenge for many, many years that I can speak of personally. Importantly, it's not alone. There is a backup series as well. We're working, of course, together and in partnership with Oncodesign on that backup series, just in case the first-in-human studies give us some cause for concern about the first candidate, maybe some optimization we can make around the PK in terms of the profile of the best drug that we can take, obviously, to the market ultimately. It is a risk mitigation, I think it's fair to say. The potential for a PET ligand, very exciting. Again, something we are working on with Oncodesign. This will give us that direct measure of target engagement in the brain, something the field has been looking for a long time. Again, very, very challenging due to the particular profile of molecule that you need to be able to measure that in the brain. So again, I think we are in good hands in working with Oncodesign to try and find a good PET ligand. Let's see where we can go. So regulatory tox studies are due to begin very soon. We have phase 1 clinical studies planned to begin next summer. It's important to say there is competition in this field. Denali, originally Genentech, have a series of molecules that has now turned into a lead molecule, DNL151. Biogen have taken over this and put some money into it, a lot of money into it. Their pivotal studies start later this year. What's important to say about that is we are, first of all, confident we can be superior to that molecule. And second of all, it gives us, again, confidence in the validation of this target, both in the ability to measure an impact in humans and also in patients as well. So we follow it as fast as we can, of course. The last thing I'll say on LRRK2 specifically is around the patient engagement. Clearly, we are very, very busy and active with European Parkinson's disease associations over in the US with Michael J. Fox and others to, of course, get the engagement of patients that we need to have a successful clinical development and to understand exactly how we position the product as we move forward. Okay, a last word from me is just to talk about our new research centre. So it's a little bit of self-promotion, but in the Saclay region, of course, neighbours to the facility of Oncodesign there, we are finalising construction of a new research and development institute. We're very, very, very excited about it. It has an incubator associated to it, and we will be the only international pharma in this area. So again, joining Oncodesign in a very, very, very important area of France for new medicines. Okay, I'll leave it there. Thanks for your time. I hope it was clear en anglais.